Good morning from welcome to all the participants in today's international webinar on the desert's fierce warrior, the camel spider, biology and research organized by BioLogic. Camel spiders are found in many parts in the Middle East, Africa, and some parts of the United States. We have received an overwhelming response from interested people, those who really want to know about the camel spider. Participants of today's webinar are from different parts of India and different countries like USA, Canada, United Kingdom, Mexico, Belgium, Israel, Syria, Austria, Czech Republic, Colombo, Bolivia. And they are from different disciplines such as life sciences, botany, environmental sciences, biochemistry, biotechnology, physiology, wildlife, and to name a few. Now, I would like to hand over to invite Dr. Sandeep Pal, Assistant Professor of Zoology, Bharatiya Rashtraguru Srinath College, and the founder admin of Biologic Academic Platform, to deliver the welcome note and to introduce today's invited guest speaker. Sir, please.
Thank you, Risha. So on behalf of BioLogic, I welcome all the participants once again. Now it's time to introduce today's guest speaker. Dr. Paula Cushing is Senior Curator of Invertebrate Zoology. She is an evolutionary biologist who studies evolutionary patterns and processes in Arachnida, that is, spiders and their kins. Her research focuses on the diversity of spiders. She is also involved with projects exploring the taxonomy, systematics, biogeography, and natural history of spiders, scorpions, and solifuges. She has published on myrmecophilic spiders, that is, spiders that live inside ant colonies, on which she delivered a wonderful talk on Biologic Platform in 2020. She is also coordinating the collections and research efforts of several museum volunteers. She is very much involved with the American Arachnological Society and the International Society of Arachnology. As we gather here in India to celebrate the dawn of knowledge, Dr. Cushing will share her expertise and research during the tranquil night in the United States. It's a wonderful demonstration of how knowledge knows no borders, transcending time zones and continents. Madam, we are honored to have you as our invited speaker. And now the virtual stage is all yours. Thank you.
Thank you so much, Dr. Sandeep Paul. Thank you, Risha. Good morning to everyone in India, and good evening to everyone in the United States, and whatever part of the day it is, wherever you are. So as Dr. Sandeep Paul said, I am an arachnologist. I am a curator, a scientist at the Denver Museum of Nature and Science in the United States. And today I'm going to give you a presentation about some of the research my lab has done on camel spiders. So without further ado, I will start, or hopefully start, my presentation. I just have to find my presentation to share it. And I don't know how to do it, so there you go.
Yes.
No.
Ma'am, just present your PowerPoint presentation. Yes. Yep.
There we go.
Yes, fine.
Okay. So sorry about that. So today I'm going to give you an overview about Solifugae. Solifugae are commonly called camel spiders, and they are in the kingdom Animalia. They're animals. They're in a phylum called the phylum Arthropoda. And arthropods include, as many people in this audience know, they include anything with an outside exoskeleton and jointed legs. So that big phylum includes insects, crabs, millipedes, centipedes, and it includes arachnids. The class Arachnida includes 12 different orders of organisms. Solifugae are one of those 12 orders. Solifugae, the camel spiders, and I'll use camel spiders and Solifugae in this presentation interchangeably, but they are found in xeric, dry, desert habitats throughout the world. They're very common predators. The only places they're not found is Antarctica. They are not found in Australia, but they're found in other habitats, other dry, xeric, desert habitats everywhere else in the world, including North and Central and South America, including in India. Now, how Solifugae, how camel spiders are related to those other orders of arachnids is still yet to be determined. So there have been some phylogenetic analyses looking at the relationships between different orders of arachnids. Some of these other studies have suggested or hypothesized that Solifugae are most closely related to pseudoscorpions. Other studies, so a couple of studies show them as closely related to pseudoscorpions. One study showed them as most closely related to an arachnid group called the palpigrades. Another study showed them as closely related to another order called the Raciniolii. And yet another study, several studies have shown Solifugae as being most closely related to different kinds of acari, mites and ticks. So we still don't know about who they're most closely related to. That is still to be determined. As I mentioned, there are 12 different families in the order Solifugae, and there are two families, the Amitrechidae and Aremobatidae, that are found in North America. My lab is focused on the biology, the phylogeny, the taxonomy of one of those families, of the Aremobatidae. But in this next slide, in green, there are three families that are found in India: Dacids, the Galeotidae, and Rhagotidae. So there are Solifugaes even in different parts of India as well. As I mentioned, my lab has been studying these arachnids for many, many years, for a couple of decades. In 2007, my colleagues and I got a big grant, a National Science Foundation grant, to study the worldwide distribution of camel spiders and to try to better understand how the different families are related to one another, try to understand more about the biology in my lab, the biology of the Aremobatidae, that one family. And then in 2018, I received another colleague and I received a second National Science Foundation grant. And this was focused on that one family, Aremobatidae. Now, through both of these grants, we've been able to recruit early career scientists to study these organisms. And I'm not sure, I think you can see my arrow. These are two of my graduate students, Erica Garcia and Goran Shikak. And Erica is no longer my graduate student because she finished her PhD recently and has been recruited to do postdoctoral studies on Old World Solifugids in Israel. Another one of my students is shown here. This is Ryan Jones, my current PhD student, and Goran is my master's student. And we have encouraged and brought into this field of study students from Mexico, other students from around the world. This is a postdoctoral student from Germany who's now a professional scientist. And this is a former PhD student of mine. So we've recruited people to start studying this group of organisms. Prior to the grants, and all of the work that my lab has done is built upon the foundational research of these two gentlemen shown here, Dr. Martin Muma and Mr. Jack Brookhart. Jack Brookhart received his master's degree from Martin Muma. Martin has passed away. Jack Brookhart is still very active in this field of study. And Jack comes into my lab and works in my lab one day a week. And Jack has been very, very involved in training the next generation of scientists to study this group of organisms. Jack has been publishing on Solifugids since the 1960s. So these grants that I received along with my colleagues were one mechanism to bring life back into the study of Solifugae, the study of camel spiders, by recruiting students and getting people excited about the research. So some of the projects that my lab has been focused on include trying to better understand the phylogeny, the evolutionary relationships between different genera. Genera is the plural of the word genus: kingdom, phylum, class, order, family, genus, species, genera. So trying to understand the evolutionary relationships among the different genera and different species within the family Aremabatidae. And we've been doing this and testing these relationships using largely DNA, so largely sequencing data, DNA sequencing data, but also we've been doing research on their morphology, different morphological or anatomical characters. Using the phylogenetic hypotheses that we have generated in the lab, we have then started to do some taxonomic revisions of some of the groups within the family, some of the genera and genera within the family Aremabatidae. We've also done research on different morphological characteristics, morphology within the order, and we've done some work and some publications on their behavior, on various aspects of their biology. And so I'm going to touch on in this presentation some of this research that we've been doing in my lab on camel spiders in the last 20 years. Now, to understand some of the research that we have done, you have to understand a little bit about the biology and the natural history of these organisms. So Solifugae, camel spiders, are major predators, as are most arachnids. They're predators of insects. So Solifugae are very, very fast-moving, very active predators of insects in these desert habitats. This picture shows a solifuge, a camel spider feeding on some termites, but they will eat whatever they can capture. And they will grab the prey using their legs and their pedipalps. And we'll talk about the anatomy of camel spiders in a moment. Then they will bring that prey close to their jaws. Their jaws are called chelicerae, and they'll tear that prey apart. And they'll regurgitate digestive enzymes onto the prey and then suck up that pre-digested liquefied nutrient bath. But they're very active predators, very pugnacious. They will try to attack whatever they think that they can eat. What we think they do in these desert environments, most of the species are active in the evening at night when the temperatures are cooler, because it's far too hot during the day for these arthropods, these arachnids to be active. So most solifugids are night active. They're coming out at night. And during the day, we believe that they spend most of their time in underground burrows. So this is a video that was taken by one of my former graduate students, Dr. Erica Garcia, and this video shows how they construct their burrow. So this is a solifugae, a female that's emerging and digging out the soil from her burrow. She's using her front legs, she's using her pedipalps, and she's using her jaws to push that soil out of the burrow. We'll watch it one more time. Now, whether or not solifugae build permanent burrows that they return to every evening, we don't know. There are many aspects about their natural history that we do not know yet. So we don't know if they have permanent burrows. We don't know if they return to those burrows every evening or if they construct a new burrow every night. We suspect that females probably will build a semi-permanent burrow when they are reproductively active, when they are ready to lay eggs. What I'm showing you in this slide is this very exciting paper that just came out this year, and it was led by the lab of my colleague Prashant Sharma and his PhD student Siddharth Kulkarni. And you can see many of these names that I've mentioned, like mine and Erica's and Ryan's names, my students' names, in this paper. So what my colleagues' lab did, what Siddharth and Prashant Sharma did, is they used next generation sequencing DNA technology to explore the phylogeny, the evolutionary relationship among the 12 different families in the order Solifugae to see how the different families were related to one another and to test whether each of the family was a good family, a monophyletic family. And what they found in this analysis is that some of the families are well-supported monophyletic groups, like the Galeotidae here, but some of them, the Aremabatidae, our family, but some of them, the families shown in some of the other families, I'll just say, like the Amatrechidae, are coming out in different, those genera are coming out in different parts of the tree, as are the members of the family Dacidae. So there are some families that do not seem to be good biological entities. They do not seem to be good monophyletic taxa. But this is a really fantastic piece of work that gives us some information about what we need to do going forward to better understand the relationships among the 12 different families of Solifugae. In 2015, my lab published used multi-locus DNA sequencing techniques to publish the first ever molecular phylogenetic analysis of any of the 12 families. So we published an analysis using DNA sequencing of the family Aremabatidae. And the hypotheses we were testing in this analysis were whether or not the genera, there are 8 genera within the family Aremabatidae, 8 different genus groups. And we were testing whether those 8 different genera were well-supported monophyletic clades, evolutionary lineages, or not. And what we found is that some of the genera were well-supported clades, like fairly well-supported, like this genus Chambria shown in orange here, and the genus Aremacosta and the genus Aremarax. But some of the other genera, such as the genera that is marked in blue, Hemerotreca, those species in the genus Hemerotreca are coming out all over the tree. So that is not a monophyletic genus. The genus Aremabades is a huge genus that includes many different species, and you can see that that green color is also coming up all over the tree. So it is also not a monophyletic genus. So this also gives us some good starting point for future tests, future analyses. And one of the things my students are doing right now is they are using next generation sequencing techniques to try to better understand next generation sequencing. I won't go into it, but it allows us to increase the number of species that we can use for this kind of DNA analysis. And they are using this next generation sequencing to try to unravel some of these taxonomic problems with these genera that are not monophyletic groups. So hopefully in the future, we will be able to do some taxonomic revisions and better understand really what are some new genera that should be defined, because clearly Aremabades, for example, is a catch-all, is a dumping ground. So as I mentioned, my lab also has done some research on different morphologies, different anatomies, anatomical, morphological characters. So in order for you to understand some of these studies that we've done, you have to understand the general body plan of a camel spider. So camel spiders, like most other arachnids, have 2 major body parts. Their head region of a solifuge or a camel spider is called the propeltidium. That's analogous to the cephalothorax of other arachnids. The second major body part is called the abdomen. So the propeltidium and the abdomen. This arachnid order, Solifugae, like all other arachnids, have 8 legs, 4 pairs of legs. They have a pair of front appendages called pedipalps, and they have very powerful jaws called chelicerae. So 4 pair of legs, 2 body parts, a pair of front appendages called pedipalps, and the jaws called chelicerae. So you need to understand that in order to understand some of the studies that we've done on their morphology. Because Solifugae, camel spiders, have some really interesting morphological structures on their body. For example, there are many different characters that are associated with their chelicerae. And that is for a specific reason, because the male chelicerae, the male Solifugids or camel spiders, use their chelicerae to transfer the sperm into the female's body. Male Solifugae, male camel spiders do not have a penis. Rather, they will deposit the sperm packet directly onto the female's genital opening, and then they push that sperm packet into the female using their jaws, using their chelicerae. So there's a lot of characters that are associated with the chelicerae that are important. They have these structures called malleoli on the underside. Males and females have malleoli. They look like little leaves on the underside of their fourth pair of legs, and they drag those malleoli along the ground as they're moving. If members of the audience know anything about scorpions, scorpions have pectines, have these structures that they drag on the ground. And pectines and the malleoli are thought not to be analogous structures. They're not at all analogous, but or homologous, but they function very similarly, because scorpion pectines and the malleoli of the camel spiders are thought to function as chemoreceptors, picking up chemical cues in the environment. Some male camel spiders have these structures, these specialized setae on the underside of their abdomen that are called ctenidia. And I'll talk a little bit about the ctenidia. On the pedipalps of the camel spiders, we see a pedipalp here. These are pedipalps. There are specialized spines, specialized hairs that are very important in the biology of these animals. So my lab has also explored and done studies on some of these different morphological characteristics as well. So let's talk first about the chelicerae. I mentioned that male chelicerae are very specialized for transferring sperm into the female. This slide here shows two male chelicerae on the left and the analogous females on the right. And what you should notice is that in the family Arachnida, females have teeth on both the upper finger, the fixed finger of the jaw of the chelicerae, and teeth on what's called the movable finger. Whereas the males do not have teeth on the fixed finger, the upper part of the jaw. And sometimes they have very strange patternings, very strange shapes to the male chelicerae that are very different than what we see in the female. In the old world, other families of Solifugae, we see these structures on the male chelicerae that are called the flagellum. And we see crazy forms in the male chelicerae in some of these other families. This is a plate from a publication by one of my former graduate students, Tarina Bird, that shows all of these really strange looking, differently shaped flagella on the male chelicerae of different species of Solifugids. So these structures, these flagella, we don't know what the function is, but only the males have them, and they must have something to do with copulation. Now, why do they have these structures? It's because the males use the chelicerae for sperm transfer. So this is an image showing a male that has his chelicerae. He's not eating the female. He's not biting her. He is got his chelicerae embedded into her genital opening, and he is pushing the sperm packet in or preparing her for receiving the sperm packet. And in order to understand what's going on, I'll show you a video by another student of mine, former student Jen Rousell. So Jen, for her master's degree, she did a mating study of Solifugids, and she gave us this really wonderful mating video. This is a female that you see here in the video. She is in a terrarium with a mirror on the bottom. So Jen was able to see what was going on underneath the Solifugae. And as we run this video, the female is moving around. She's very active. She's moving. And then suddenly the male is going to run at her. The male grabs her. He holds on to her with his legs, and look at the pedipalps of the males. He's using the pedipalps to manipulate and maneuver her body. And as soon as he makes contact with the female, she goes quiescent. She stops moving. So she becomes completely lethargic. This is the female. Her body is bent over. The male is maneuvering her body using his pedipalps, and we'll talk about what exactly he's using when he's doing this. And he's chewing on her. He's stuck his chelicerae into her gonopore, into her genital opening. Now, we don't know what he's doing here. We hypothesize that he is either preparing her internal anatomy to receive his sperm packet, or he may be removing the sperm of competing males. We don't know. Or he may be doing both. But he will use his chelicerae and chew. It looks like he's chewing her for quite a while. This goes on for several minutes. All the time, she is completely quiescent. And look at his pedipalps. He's using those pedipalps, and specifically, he's using these organs called suctoral organs, which I'll talk about in a moment, to hold on to her body and to move her abdomen to and fro. Now, he is about to remove his chelicerae. Now, right there, he moved his body up and he deposited the sperm packet right onto the female's genital opening. Then once the sperm packet has been deposited, it's very rapid. He then pushes the sperm packet into her body using his chelicerae, and then he continues to chew on her or to move his chelicerae while he's got them inserted into her genital opening. Watch the female. As soon as he has pushed the sperm packet into her, she begins to move. She begins to wake up. And this goes on for quite a while. This goes on for a few more minutes. We're just going to skip ahead because he continues to chew on her. She continues to wake up until finally she's had enough, and she will try to escape from his grasp until finally he lets go of her and they separate. There we go. So the male is using his chelicerae to insert this, to prepare her for receiving the sperm. Then he deposits the sperm onto her opening with an abdominal contact, abdomen to abdomen contact. And then he uses his jaws to push that sperm into her again. Kind of disturbing, but that's that's sex in Solifugae. That's how they copulate. Once copulation happens, the female, we don't know how she releases the sperm to fertilize the eggs, but fertilization occurs probably as the eggs are leaving her body, but we're not quite sure. On the upper left, you see a female that is in the process of laying her eggs. What happens after she lays her eggs? We hypothesize that she lays her eggs in these underground burrows. We hypothesize that she will stay with and guard those eggs, but she may die. Most females may die after they lay their eggs. We really don't know much about that part of their natural history. But the eggs, once they're laid, take a week or so to hatch. And when the egg hatches, when the larvae emerges, the larvae are very helpless. They will all aggregate together, and they might twitch a little bit, but they can't really move. So they're all staying in an aggregation together until they go through their first post-embryo molt. So when they molt again, after emerging from the egg, they molt. And then we begin to see in the bottom left, we begin to see a little bit of color and a little sclerotization, hardening of the jaws. But they're still not particularly active. And it's not until they go through one more molt and enter the second instar nymph, the second and third instar nymphs, that we begin to see a hardening of the jaws and more color patterns apparent in the body. and a lot more activity. And at that point, by the second, third instar, they are active and they are hunting. And at that point, they will start cannibalizing each other. So we think that about that point in nature is when they will start to disperse, because otherwise they will be food for brothers and sisters. So that's all we know about their development. I hope that someone out there is interested enough to raise some. We don't know how long it takes them to develop to maturity. We don't know how many instars they go through. There's a lot of aspects about their natural history and biology that remain to be determined, remain to be studied. I talked about the malleoli, these really interesting organs that are found on the underside of the legs of both males and females. This is a close-up view of a malleoli in the bottom right. And the malleoli, a really nice paper came out in the 1970s by a couple of researchers who demonstrated that the malleoli function as chemoreceptors. And we think that the Solifugae are using the malleoli to detect chemical signals from each other and from prey that may be around. So it may be a way for them to detect when there's an insect around that they can eat. And I think there's some behavioral support for that hypothesis. Another student was doing some field research with me, Kyle Conrad, when he noticed that there were all these little juvenile, the little third instar Solifugae that were running around in this sand dune area. And as Kyle watched, he noticed that the little baby Solifugae would run, and then they would stop, and they would back up, and then they would start digging in the sand. And sure enough, there was an insect beneath the sand, and they were digging up that insect to eat it. It may be that they were picking up the chemical cues of that insect underneath the sand using those malleoli. I talked about, when I showed you the video of copulation, I mentioned how the males were using these organs called the suctoral organs to hold on to the female's body. Male and female sulfugids of all the families have these suctoral organs that they can evert, they can pop out of the tip of the pedipalps, and they can pull those organs back in. And those suctoral organs, this is a scanning electron micrograph of a suctoral organ. It looks like a little balloon structure when they evert it, when they push it out of the pedipalp. And it functions kind of like the toe pad of a gecko. So it functions in holding on to smooth surfaces using hydrophilic forces. So Solifugae, for example, camel spiders can climb up the glass wall of a terrarium using just those suctoral organs. They use the suctoral organs, the males do, to hold on and manipulate the female's body when they're copulating with her. The Solifugae use those suctoral organs to pull prey closer to their chelicerae. So we did a study in my lab looking at the functional significance of these suctoral organs. This is a scanning electron micrograph photo of an everted suctoral organ. And you can see that the surface is variegated surface. It's holding on, it's forming a hydrophilic bond with a very fine surface of water that's on smooth surfaces. So in this study that we published in 2005, and a colleague did a much nicer study, Anya Klan, repeating some of the work that we had done, but hers is really beautiful, her 2008 paper. But in both of these papers, we demonstrated that the suctoral organ would be pushed out of the pedipalp, of the tip of the pedipalp, through an increase in hemolymph or blood pressure, and it would be pulled back in using tendons. So the animal could pull them in back into the pedipalp using these tendons. We also did some analyses and some studies of the specialized CD that are found on the pedipalps of the different families of camel spiders. So a student, Patrick Casto, and I published a little paper where we did a really simple survey using scanning electron microscopy of the various kinds of CD that are found on the pedipalps of each of the 12 families of Solifugae. And one of the things we found is that these CD that had a bifurcated, a fork-like tip are found in all 12 families. But there are some CD that we only saw in one or two of the families. So we were just trying to get a feel for what some of these sensory CD look like. We also did some studies of these specialized CD called papillae. Papillae are specialized CD found on the underside of male pedipalps of 3 different families. So this is looking at the underside of a pedipalp at a field of these papillae. And the 3 families in which we see these specialized CD are the Arachnida, Cartiidae, and Solifugae. And you can see that in each of these 3 families, the papillae look quite different. So there's a family, they are diagnostic at the family level. So Cartiidae papillae look very different from Arachnida papillae, look very different from Solifugae papillae. But we wanted to find out what was their functional significance of these really interesting, beautiful little specialized CD. So we used different techniques, transmission electron microscopy, SEM, scanning electron microscopy, electrophysiology, and just light microscopy to test different functional hypotheses about the function of these specialized CD. So we hypothesized that maybe they were mechanoreceptors detecting movement in the environment. Maybe they were contact chemoreceptors or olfactory receptors picking up chemical cues from the environment. Maybe they were secretory organs producing some kind of a chemical signal. They're only found in males, so maybe they had a sexual signaling function. So again, we use different methodologies to test these hypotheses. If these structures, for example, were mechanoreceptors, then this is looking at a cross section through one of these little Christmas tree looking papillae. If they functioned as mechanoreceptors, in arthropods, mechanoreceptors should have a tubular body, a dendrite that ended at the base of the CD. If it functioned as a olfactory or chemoreceptor, it should have a dendrite that extends through the shaft of the CD. And if it functioned as olfactory or chemoreceptor, it should have pores, openings to the outside. So electrophysiology was another technique that we used with live Solifugae, in which we exposed a live Solifugae with papillae to different chemical signals and to movements and to heat and to humidity to test the functional significance of these papillae. What we found is that we did find evidence that there were dendrites, there were nerves that extended through the shaft of the papillae. We found evidence of these tubular bodies that are characteristic of mechanoreceptors at the base of the papillae. And this is what we think the structure looks like. We think that it has a dendrite that extends through the shaft. We think that it has tubular bodies at the base. We saw pores in the openings to these little branches. So those are all structures, microscopic structures that we would expect of a chemoreceptor. So there's evidence that these are chemoreceptors picking up chemical cues. Some of the other experiments that we carried out provided no support for thermoreception, for them picking up heat signals, no support for hygroreception, no support for secretory function. We saw no glands at the base of the CD, but we did see support that they may function as chemoreceptors. So it may be that the males are using these papillae to pick up chemical signals from the female. So this was a study that we published in my lab in 2014. My student Erica Garcia, Dr. Garcia now, did a survey of the spines on the pedipalps of Solifugae to look at how different the functional significance of the bases of these spines may be. So in some cases, in some genera, we see spines that seem to be more movable, that seem to be able to move back and forth more so than the spines on the pedipalps of other genera. Like the genus Aremarax, their spines don't seem to be movable, whereas in the genus Horibades and Aremachilus, they have these notches in the bases of the spines that allow the spines to be more mobile. My student Ryan Jones published a paper in 2021 in which he was trying to determine the phylogenetic significance of these specialized CD on the underside of male abdomens that are called ctenidia. And we use the ctenidia as diagnostic characteristics to distinguish between different genera. We use the number of ctenidia to differentiate between different species within a genus. And Ryan did a nice study where he was testing whether the number, the length, and the shape of the ctenidia had phylogenetic informativeness. And what he found was that the number has a phylogenetic signal. It's important whether they have ctenidia or not. Some genera don't have ctenidia at all, and that seems to be evolutionarily meaningful. But the length and the shape tend not to have a significant signal. So you can have various numbers of ctenidia, various shapes within, sometimes even within a species. We've also done some studies looking at how to effectively capture Solifugae, and we published some papers on the effective ways that scientists can use in the field to find and collect sulfugids. So what we've known for a long time is that these night active predators are attracted to lights at night. They're attracted to illumination. And so what we do now is we use pitfall traps, which are basically just containers with preservative. We use propylene glycol as our preservative, and we hang lights over our pitfall traps in the field. We turn the lights on, and we find that the lights attract the sulfugids to the area of our pitfall traps and maximize our chances of getting sulfugids falling into these cups with the preservative. So we use light trapping as a way to collect sulfugids in the field. And as I mentioned in the beginning, one of the great things about our research that's been going on in our lab is it has been an exciting way to get and attract students into this field of biology. So there's a lot of students out there. My colleague Edmundo Gonzalez Santillan in Mexico has recruited a couple of students, Jair Castillo and Diana Batista, into this field of research. So they're doing projects on sulfugids. Erica is going off to Israel to do her postdoc on rhagodids, on other families that are found in Israel, other families of sulfugids. Ryan and my student Goran are finishing their degrees studies in my lab. Carlos Santabañas Lopez, we supported as a postdoctoral student, and he is now a professor at Eastern Connecticut State University. So we've attracted and gotten a lot of students involved in this research, which has been really fun. We've also developed a website. So if you go to www.solifugae.info, my colleague Warren Savory has been really instrumental in developing this website, which provides a lot of basic information about this order of arachnids and about their biology, what we know about their biology, their taxonomy, their biogeography, and provides a lot of bibliographic information as well. So this is a really great resource for people who are interested in this order. And in my lab, we're starting to develop a field guide, an online field guide to the species within the family Aremabatidae. And this online field guide is available through the Scan Symbiota database. And we are providing diagnostic images that you would need to know, you would need to see in order to identify sulfugids within the family Aremabatidae down to species. And through this website, we're also providing interactive maps. And in these interactive maps, these little dots, you can click on any of these dots and it will take you directly to the database record for that particular specimen. And it's a great, exciting time to go into the studies of camel spiders. There's a lot of papers coming out from labs all over the world that are looking at the taxonomy of this group of organisms. They're looking at and exploring their biology, their mating behaviors, information and studies about their anatomy and their morphology. So there's lots of really interesting papers that are coming out all the time about this group of camel spiders from labs all over the world. And with that, I'm going to thank the audience for tuning in tonight. And I am going to stop sharing so that I can get back and I can see about answering some questions from the audience. Dr. Sandeep, I think you're muted.
Yes. Thank you, ma'am. Thank you, ma'am, for your wonderful deliberations. And now it's time for question answer session. So let's see what are the questions we have in comment section. So this is the first question from Julie Oatfield. That is, hi, are the sartorial organs analogous, evolutionary, to how spider pedipalps are used in reproduction, or totally different? Surprised to learn Solifugae use their chelicerae for sperm transfer.
I know the use of chelicerae for sperm transfer is a little bit disturbing to me, but yes, that's what they do. In terms of the septal organs are a unique structure found only in camel spiders, and the pedipalps of the pedipalps in general of Solifugae are homologous to the pedipalps of all the other arachnid groups, but in each of those different orders, you see different structures and different functions for those front appendages, for the pedipalps. So with spiders, for example, the spiders have the males have very specialized pedipalps that they use for sperm transfer, whereas in Solifugae, those pedipalps are not what's used for sperm transfer. That's the chelicerae are used for sperm transfer, but the pedipalps in Solifugae are used to hold on to the female.
Okay, thank you, ma'am. And there is another question from David. Ma'am, showing you. Ma'am, earlier you mentioned that during the process of copulation, the male spider takes hold of the female spider and the female spider becomes quiescent. Does this phenomenon occur due to some kind of chemical interactions between the two individuals?
I wish we knew. That is such a great question, and we have no idea. We hypothesize that the male's physical contact of the female with his chelicerae, when he pinches her body with his jaws, it's that contact, it's that physical contact that is triggering the female to stop moving. But we're not certain. And it could be that there are some chemical cues that are also used. And we also don't know why is the female waking up all of a sudden? So we don't know anything about that process. We don't know what's triggering the female to go quiescent. We don't know what's triggering her to come out of that state of lethargy.
Okay. Next question is, what are the roles of malleoli in sexual behavior, specifically mate selection?
Another very good question. No, and there's so many aspects of the biology of these arachnids that we don't know yet, that they're wide open for researchers to explore. The malleoli, it has been demonstrated that they are chemoreceptors, so it certainly could be that the males can pick up some sort of chemical signals that the female may have deposited in the surface of the soil. It certainly be that he is able to do that using the malleoli, but we don't know for sure. And it's not just the males that have the malleoli. Males and females both have those structures.
Okay. Thank you, ma'am. Next question is from Julian. Thank you for the talk. Solifugae are fascinating. How could we check the function of the flagellum on the chelicerae yet for sure?
Such good questions. All such good questions. And that is another big question in sulfugent biology. We have no idea what the function of those flagella structures are. They're so strange. And they're only found in the males, so they certainly must have some kind of function in sperm transfer or titillating the female or something. But maybe somebody in the audience, some students out there, are the ones who are going to figure that out.
Yes. There are so many questions. Next question is from Shreya Poddar. Why do you think the mother dies after laying the eggs? Is she eaten by the babies?
Yeah, we don't actually know. She may not die. She may survive. We're not sure what's going on inside that burrow. We think that the female will guard the eggs for a while, but we don't really know. It could be that she dies. We have no idea what's going on. In captivity, they don't live very long in captivity. So that series of photographs I showed you of the female laying eggs and the juveniles, I felt really lucky to be able to take those photographs, because usually you bring a female into the lab, and if she lays eggs, you're lucky if the eggs hatch. Usually they don't hatch. You're lucky if the female lives long enough in the lab to even lay the eggs. And you're really lucky if the eggs hatch and the juveniles survive for a few molts. So we really don't know much about how long the females live. We think that at least with the Arachnida, the family that we study, we suspect that the whole lifespan of males and females is probably only a year, maybe a little over a year. So we don't think that they have multi-year lifespans, but we don't know.
And there are still so many questions unanswered in science, right?
So many questions. We need more enthusiastic young scientists to go into these fields.
Next question, ma'am, from Miguel. Hi, thanks for the marvelous talk, Dr. Cushing. How difficult it's been to conciliate morphology with the information that molecular phylogenetics have provided?
Excellent question. That is our major challenge now. So my students are beginning to finalize the phylogenies resulting from the DNA data. And now what we're trying to do is exactly that. We're trying to look at these well-supported lineages, these well-supported clades, and we're trying to figure out, are there morphological characters that we can use that will differentiate that clade, that lineage from any other lineage. And it's a real challenge. There's a really great researcher, Hernan Iuri, down in Argentina, who's doing a lot of really interesting studies of the different morphological characters of Solifugae. So he's very likely to come up with some informative morphological characters that can be used to match onto or to differentiate different evolutionary clades. That's a great question.
Next, move on to the next question. My question is, if we do not know about the name of behavior of any particular spider, so how can we find the particular behavior terminology?
Now, that's a good question. So I think what we do know is that arachnids, the class Arachnida itself, is well supported. So we know that we don't know which arachnid group is most closely related to camel spiders, but we know that all the arachnids evolved from a common ancestor. And because we know that, then a lot of the behavioral ecologists, some of the behaviors that have been demonstrated and the behavioral terminology that has been used for, let's say, the Arachnida, for the spiders, may be useful and may be applicable with other orders of arachnids because they evolved from a common ancestor 300 million years ago. But the terminology and standardization of terminology it's a good point because it can be really confusing if you're looking at a behavior that looks very similar in Solifugae. It looks similar to a behavior in a different order, and the behavioral ecologists are using different terminology for those different groups. So you want to use the same terminology, but for each of the orders, there's always going to be some discontinuity between the terminologies used. I'm not sure if I quite answered that question, but
Yes. Thank you, ma'am. And next question is very interesting. This is from Faizi Ali Khan. Actually, ma'am, we discussed. I question: is there any Solifugae, that is camel spider, in India? But you said yes, there are a few species of camel spider, right? So here goes the question. Hi, Dr. Cushing, great talk. I recently found a Solifugae specimen from peninsular India. I suspect it to be a member of Ragobiti. Yes. But I really want to confirm the species. But I have yet get my hands on a comprehensive guide for that. Do you have some suggestions?
I think the best place to start is that www.solifugae.info site, and that will at least give you an idea that of a starting point. And that site also has a lot of bibliographic references. So if you can figure it probably is a rag-coded, so you're probably right about that. And in terms of how to key it to the genus, that's always going to be a challenge because a lot of the literature is very old. But that website is where you should start.
Okay. Next question is from Shomo Mondol. In case of evolution, we see they transfer egg by Chelsea many times. It rupture gonophore so that the number of these species are become decline. What about this?
Well, they're not transferring eggs by the chelicerae. They're transferring the sperm. I think the person meant they transfer the sperm using the chelicerae. I don't think it's rupturing the gonopore. It's inserting into that opening. And we think what may be happening when the male chews on the female before transferring the sperm is he may be preparing her for receiving the sperm. But we're not seeing damage, or I don't see a lot of damage to the gonopore. There's at least one study where our colleagues have shown that there can be damage. So there can be some damage to the chitin surrounding the female's genital opening, but it's not terminal damage. So the male, when he pushes his chelicerae in, it may do a little bit of damage, but she's not losing so much hemolymph that she would die.
Uh, Next question is from Nusrat Mazid. A nice talk, Dr. Paula. Please guide about the selection of outgroup for phylogenetic tree construction.
Sure. So it depends. It depends on whether on what level you're trying to look at. So my colleagues' phylogeny of all 12 families, Siddharth Kulkarni and Prashant Sharma, that study where they were testing the relationships between and among the 12 families of Solifugae, they would use a group outside of that order. So they used, and the way they would decide upon the outgroup is based upon other colleague studies. So they would use a pseudoscorpion, a ricinuleid, an acarine, because in those other phylogenetic analyses, those are the most common hypotheses as to the orders that were most closely related to sulfugids. So they might use those one or all three of those representatives as outgroups for the sulfugid level phylogeny, the order level phylogeny. When we're doing our phylogenetic analysis of the family of Aremabatidae, we can now use Siddharth's phylogeny to figure out what is hypothetically, what is their hypothesized sister family to the Aremabatids. And we would use that sister family as our outgroup, as well as one that's more distantly related. So we might, in our analysis in 2015, we had no idea. We didn't know which families were most closely related to Aremabatidae. So we used 3 different families as outgroups.
Okay. Next question is from Rupam. A wonderful talk, ma'am. You said the spiderlings may have cannibalism. So, what is the evolutionary view regarding this kind of solitary strategy for living to most of the spiders?
Yeah, for the majority of arachnids, and then this is true for the majority of spiders. Spiders are in the order Araneae. Solifugae might be called camel spiders, but they're not real spiders. For camel spiders and for spiders and most scorpions, these are all predators. They're predatory arachnids. All of them will readily eat each other. So cannibalism is pretty high in most of the arachnid orders, in the Solifugae, Araneae, Scorpiones, and that is that's a consequence of being a primary predatory organism. So you are right. The questioner was correct that most of them are living solitary livers. They live separate from one another unless they're coming together to mate.
Okay. Next question is from Bilal Shahid. He is from Pakistan. Hi, Professor. Is there any mechanism that you have adopted to check the behavior of spiders from social to solitary, from spiderlings to adult one?
The Solifugae are not a single one of the species of Solifugae have been documented to be social or semi-social. So where you see sociality in arachnids is really only with some groups of spiders, as far as I know. And where we see sociality or social behavior in spiders and Araneae, that other order of arachnids, you see it's readily apparent, because you will see spiders of the same species that are living in an aggregate group. The only time in Solifugae that we see that kind of aggregate living is with the babies, and only up to a point. After they have gone through 2 or 3 molts, then they scatter and then they are solitary.
Okay. Another question from Sravanti Chattopadhyay, that is, ma'am, does Chinidia have any special functions?
Probably, we don't know. We really have no idea what the function of those strange little CD are. They must have some function, but we don't know what they
Yeah.
are.
And ma'am, another question from Julian, that is, are some species of Solifugae endangered?
That's another very good question. I bet there are some that are threatened or endangered because of all of the habitat degradation. You know, desert systems are themselves endangered habitats. So in many parts of the world, the desert habitats are being irrigated for farming, and we're losing that very unique habitat, that desert environment. So I suspect that a lot of these desert adapted organisms, including many species of Solifugae, are being driven to extinction or being threatened by this very abrupt change in the habitat. Um, there's a lot of areas in the world where we're seeing urbanization and the habitat destruction itself, I'm sure, is affecting some of these species. However, we don't know enough about the distribution, the population structure, the behavior of this group of arachnids to know for sure which ones may be threatened or endangered.
Uh, One last question for you, ma'am. This is from Miguel again. I find interesting that for an order that has the avoid the sun implicit in the name, they are attracted by light at night. Do you have any hypothesis about that?
Such a good question. I am so sorry that for all of these really great questions, the answer is I don't know. But that question drives me crazy. Yeah, why are they attracted to light? The hypothesis that I have is when there's a lot of insects that are attracted to light. There are a lot of moths and night active insects. And I've read some studies that suggest that some of these insects are attracted to light because insects are attracted to open spaces. That's an easy area for them to fly. And so I hypothesize that Solifugae, maybe they've evolved this attraction to light for the same reason. In other words, they're attracted not to the light, but they're attracted to open spaces where they're more likely to encounter their prey. And because they have the same trigger, the same evolutionary attraction to open spaces, but for a different reason than the insects do. Insects are attracted because it's an easy flyway. Solifugae maybe are attracted because it's a good place to get prey. But I don't know. It's such a good question, and all I know is lights work.
Okay, so there are no more questions from comment section. If Risha and Rupam have further any questions, you can ask, ma'am.
Ma'am, like I want to ask, we know many animals show some courtship ritual. So do camel spider also have this courtship call or any kind of ritual for attracting the females?
Good question. I don't know. I I don't know. There's got to be a way for them to find each other.
Ma'am, any kind of pheromone or something? Like
Yeah,
I'm really curious about it.
that would be my hypothesis is that they're able to locate the female maybe using chemical signals. I don't think they have very good eyesight, so it's not like within the araneae, within the spiders, jumping spiders can see each other. I don't think that's going on here, so I don't think they're using visual cues. So they must be able to locate each other using something else. And it's probably chemical cues, but we don't really know.
Thank you.
Sure.
Ma'am, one more question from comment section. There are so many questions there when continuing to come. Ma'am, totally fascinating to that the chelicerae of arachnids could be used to sperm transfer as well. How more surprising Mother Nature could be? Okay, ma'am, question is, does the papillae, which is functioning as mechanoreceptors, are species specific? How does the males are transferring the mechanical stimuli to the females?
I don't it's another really good question. Yeah, the papillae are only found in males, so they almost certainly have some sort of a sexual function or function in finding or or titillating the female or something. Um, but we don't really know. We don't know if they are used to. We don't know. We have no idea.
And there is a connecting question. And are CT providing as a tool to inhibit interspecific mating in different arachnids?
Maybe, maybe. Again, we no idea, but that's a valid hypothesis.
Okay, so I think there are no more questions. I would like to thank all the participants, all the so many interesting participants are from around the world, and they asked so many questions and made this session so interactive. And on behalf of BioLogic, I'd like to thank Risha and Rupam for your timely assistance. And uh, Madam is just outstanding today. So,
Even if I couldn't answer any of the questions.
no, actually, there are so many questions still unanswered in science. And research is going on, and it will come out, and it is to the next generations of scientists to discover new things, right?
Yes. Oh, absolutely.
Okay, from that's all for today. So, thank you.
Thank you. Thank you very much.